I'm Kevin Esfeld, head of the Sculpting Evolution Group, where we work to advance biotechnology safely. We develop open source platforms, evolve new molecular tools, and generate data sets to empower and accelerate the work of others. One platform we develop involves liquid handling robots. Liquid handling robots are a key tool for accelerating research in biotechnology, but they're really hard to use. We developed an open source software platform that makes them really easy to program, allowing a community to share their protocols with each other and program the robots to do things that they can't do with the native software. So we use these robots to control synthetic ecosystems in which we trick fast-evolving bacteriophages to evolve useful molecules. Each well of a plate is a separate ecosystem in which a population is evolving an independent solution to that particular problem. By tracking which conditions result in better solutions, we can generate data that can be applied to machine learning programs to better design useful related molecules of that particular type. Some of our tools can be used to engineer entire populations of animals and their associated ecosystems. For example, we're working to engineer organisms to resist entire classes of pathogens, such as influenza. This could be used to help prevent future pandemics and improve the well-being of animals. The Mice Against Ticks project is an example of community-guided ecological engineering, in which we've been working with the residents of Nantucket and Martha's Vineyard to engineer the white-footed mice that are the main reservoir of Lyme disease so that they don't infect the ticks. This kind of ecological engineering project can only occur with the guidance of people who live in the area that would be affected. Because we develop technologies that can spread on their own, we're responsible for preventing accidents and misuse. One way we do that is to bring together different disciplines to ensure that only legitimate scientists have access to DNA that could cause future pandemics. And finally, we want to prepare for the worst. Imagine a future in which there's a fast-spreading HIV equivalent that spreads around the world without showing initial symptoms until most people are affected. To ensure that doesn't happen and we spot every new threat early, we're working to build a nucleic acid observatory that uses deep sequencing of wastewater to detect every new pathogen before it begins spreading across the globe. These kinds of projects that bring together different disciplines and communities can only happen at a place like the Media Lab, where together we work to build a flourishing future for humanity.